Today I'm going to talk about this thing called epistasis. Before we get into that, I'm going to go over a few things that you will need to know to fully understand this topic. First thing you need to know is a chromosome. This is a DNA molecule that carries genetic information. Next is an allele. This is something that changes one gene from another gene, and thus may impact the phenotype or physical characteristics of an organism. Next we have a locus or loci if there are more than one. This is a location on a chromosome. Now we have a genotype. This is a genetic characteristic. When we have a dominant genotype, it is typically displayed with a capital letter. A recessive genotype is typically displayed with a lowercase letter. A phenotype is a little different. It is a physical characteristic that you can see. Different genotypes can have the same phenotype. Now epistasis. The scientific definition is this: an expression of alleles at one locus masks or modifies the expression of other alleles at another locus. To make this a little simpler, we're going to only focus on two different loci. One is big A and the other is big B. If these two are put together, the big A will give the big B a mask and make it look like a big A, even though it is actually a big B. This would be an example where big A allele is epistatic to the big B allele. Whatever allele that is paired with big A will get a mask from the big A and make it look like a big A, even though it's not. There are 6 types of epistasis: single recessive epistasis, single dominant epistasis, duplicate recessive epistasis, duplicate dominant epistasis, and dominant/recessive interaction. One important thing to realize is that epistasis is an inheritance between products of different genes, and dominance is the interaction between different alleles at one gene. Now I'm going to go a little more in depth with single dominant epistasis. This type is when there is a dominant allele at one locus that will mask the expression of the alleles at a second locus. In this example, A is epistatic to B and B. If 2 heterozygous organisms mate, then there should be a ratio of 12 to 3 to 1. Here's an example we are going to use. If there is a capital A anywhere in either allele, the organism is going to be happy. If there is no capital A, but there is a capital B, the organism is going to be sad. If there are no dominant alleles, the organism will be neutral. I'm going to go over 2 different Punnett squares to show how many offspring will be happy, sad, or neutral. Looking at a capital A, lowercase capital B, lowercase b, crossed with a capital A, capital B, lowercase b, 2 heterozygous parents, you will see that 12 of the offspring will be happy because they contain a capital A. 3 will be sad because they do not contain a capital A, but they have a capital B. And then down in the bottom right corner, you will see that 1 is neutral because they do not have a capital A or a capital B. Lastly, we are going to go over another cross, this time with capital A, lowercase a, and 2 capital B's, and lowercase a, a capital B, and lowercase b. As you see on the Punnett square, there is going to be 8 happy offspring and 8 sad offspring. This will be no neutral offspring because there is no situation where a homozygous recessive offspring will come from these parents.